Hello friends, in this video I'm going to start with this very important topic, glycolysis. I'll be taking glycolysis pathway in a story form so that you are not cramming all the steps. In this story, you will be knowing that why the next step is occurring. So that's why it will be very easy to understand. And do watch the basics of carbohydrate metabolism video before taking this glycolysis video, right? So let's get started. Glycolysis, also known as Embden-Meyerhof pathway by the name of scientists who have given it this, also known as EMP pathway, short form EMP pathway. So you know HMP is hexose monophosphate pathway which produces NADPH, that is a minor source, that is a minor pathway of oxidation of glucose. But glycolysis, EMP pathway is the major pathway of glucose oxidation. So even if EMP written in the option, so you should be knowing that this is glycolysis written, right? So let's start. The first step is glucose gets converted to glucose 6-phosphate. I told you the first step which occurs is always phosphorylation, and this is because of this is for the entrapment of glucose inside the cells, right? So this first step is phosphorylation, glucose to glucose 6-phosphate. Here, for adding this high energy phosphate bond, ATP is used. ATP is used, gets converted to ADP. The enzyme must be some kinase. I told you whenever something with ADP, ATP, mostly enzyme is a kinase, right? So here ATP used, enzyme is kinase. Here it is hexokinase or glucokinase. The difference between hexokinase and glucokinase we'll be later doing in enzymes topic, right? So the first step is done, hexokinase, ATP is used. Why is glycolysis occurring in cells? The reason is to obtain ATP. But at the very first step, we are using ATP. We are doing opposite of the target. Like when opposite of the target is done, this is known as rule of business. Because, see, in business, we want money, right? But at the very first step, we have to invest money. So only first when we invest, later we'll be able to gain. So glycolysis is also like a business that first we are adding ATP and after that we'll be obtaining ATP. And any good business is that where we invest less and we obtain more, right? So I'll be taking glycolysis as a rule, as a plan of business. The nature has made this plan of business. So let's see how. The second step is glucose to fructose, glucose 6-phosphate to fructose 6-phosphate. Because glucose and fructose are isomers of each other, so here the name of enzyme is isomerase, simply, right? And in the next step, fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. So here we are adding one more phosphate at position number 1 now, making fructose 1,6-bisphosphate. Phosphate is a high energy bond. To add phosphate, we always use ATP. So here also ATP used, gets converted to ADP. Enzyme must be some kinase, and that is phosphofructokinase 1. Short form, this third number step, short form PFK1, phosphofructokinase 1. This is the rate limiting enzyme of glycolysis, also the first committed step of glycolysis. What do you mean by committed step? Committed means when this step will occur, then it is very sure that glycolysis will occur. That means first and second steps of this pathway were not committed for glycolysis because glucose 6-phosphate and fructose 6-phosphate, they can be used for other pathways also. But fructose 1,6-bisphosphate, when this compound is formed, fructose 1,6-bisphosphate, then we are very sure that now glycolysis will occur, pyruvate will be formed, right? So therefore I say PFK1 is the committed step, first committed step of glycolysis, right? Now let's see the fourth number step. Fourth number step is done by enzyme aldolase A. Write down A here, aldolase A. Where is aldolase B? Aldolase B will be doing later in fructose metabolism, right? Aldolase A is in glycolysis. The substrate for aldolase A is fructose 1,6-bisphosphate, whereas the substrate for aldolase B is fructose 1-phosphate, right? Let's see what aldolase A is doing. Aldolase A will break down this compound. This is a 6-carbon compound with 2 phosphates, which is now broken down to 2 3-carbon compounds. One is aldehyde sugar, other is keto sugar. Both are given 1,1-phosphate each. So let's see. Fructose 1,6-bisphosphate gets converted to dihydroxyacetone phosphate. This is 3-carbon keto with a phosphate. dihydroxyacetone phosphate. the short form DHAP. Later I'll be using the short forms. DHAP is formed. On the other hand, 3-carbon aldehyde is glyceraldehyde 3-phosphate. So this 3-carbon aldehyde is also given 1-phosphate. So now we have got DHAP and glyceraldehyde 3-phosphate as the end products of number 4 step. Now number 5th step is DHAP also gets converted to glyceraldehyde 3-phosphate. This way we get 2 molecules of glyceraldehyde 3-phosphate, and the name of 5th number enzyme is this aldehyde and keto interconversion is always by isomerase. So number 5th enzyme is isomerase. Now see this glycolysis pathway as a whole. There are 10 steps in glycolysis. These first 5 steps we are done. These first 5 steps constitute phase 1 of glycolysis, also known as energy utilizing phase of glycolysis, or you can say investment of business. This is the investment of business, right? And phase 2, the next 10 steps come in phase 2, and that is energy producing phase of glycolysis. Energy producing phase, right? Now see one smartness planned by nature. I told you that any good business is that we invest less and we obtain more, right? Now see phase 1. In phase 1, how many molecules are entering? How many glucose are entering phase 1? Only 1 molecule of glucose is entering. And therefore, whenever we use ATP in phase 1, we have to use it for 1 molecule. But in phase 2, how many molecules are entering phase 2? 2 molecules of glyceraldehyde 3-phosphate are entering. So that in phase 2, when we obtain energy, then every energetics is multiplied by 2 because 2 molecules are entering. This is a smartness of business planned here, that investment is less but we obtain more energy. Now let's see the detail of phase 2. The first step in phase 2 is number 6 step, that is glyceraldehyde 3-phosphate gets converted to 1,3-bisphosphoglycerate. This is the master plan of glycolysis. This step is very special step in glycolysis. Understand this very carefully. See here, glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglycerate. Can you see in this step one more phosphate is coming at position number 1? This phosphate, we are adding phosphate, high energy bond. Usually to add phosphate we use ATP. But in this step we are not using ATP because we are in energy producing phase. We are in phase 2. So we will not use ATP here. So this phosphate is simply taken from inorganic phosphate. Inorganic phosphate is always there in the medium, so we can take it. But generally, I repeat, generally whenever we add phosphate we use ATP. But this step is very special step where ATP is not used but phosphate is added. This is a very special and rare step which can add phosphate without using ATP, right? And the name of this enzyme is glyceraldehyde 3-phosphate dehydrogenase, and this is the only step of glycolysis which can use inorganic phosphate. And see, the name of enzyme is dehydrogenase. I told you whenever dehydrogenase means hydrogen removed and somebody will come to take this hydrogen, that somebody most of the time is NAD. Here also it is NAD. NAD will take this hydrogen, forms NADH. I have written 2 here because we started with 2 molecules of glyceraldehyde 3-phosphate. These 2 NADH will later go into ETC to give 5 ATPs. So this step is responsible for giving 5 ATPs. See, this is such a special step. In this step, we are adding phosphate. And to add phosphate, we generally use ATP. But this step is not using ATP. In fact, it is producing ATP, and that too 5 ATPs. Till now, we have just used 2 ATPs and we have obtained 5. It is such a smart plan of business, and many more ATPs are yet to come. Now let's go further. We have reached till this point 1,3-bisphosphoglycerate. Now this 1,3-bisphosphoglycerate is a 3-carbon molecule having 2 phosphates. Now only one work is left in glycolysis, that is, we have 2 phosphates. Phosphate is a high energy bond. Now we will keep breaking these 2 phosphates one by one, and when we break high energy bond, then energy is released. That energy can be trapped in the form of ATP, right? Only this work is left. So let's see. 1,3-bisphosphoglycerate gets converted to 3-phosphoglycerate. So you can see we are removing phosphate from number 1 position. 1,3 gets converted to 3. We are removing phosphate from number 1 position. Who will take this phosphate? ADP will take this phosphate, gets converted to ATP. I have written 2 here because we started with 2 molecules. And whenever ATP formed at some step, immediately there and then only ATP formation occurs, known as substrate level phosphorylation. And enzyme of substrate level phosphorylation is always a kinase. And here it is phosphoglycerate kinase. Short form is PG kinase. PG kinase is a substrate level phosphorylation step of glycolysis. They ask you many questions on substrate level phosphorylation steps. So it is very easy to learn these steps. You have to learn the substrate, you have to learn the product, you have to learn the enzyme of such a step, right? Now we have reached at this point 3-phosphoglycerate. Now we have our last phosphate attached at number 3 position. We can remove this phosphate also and we can obtain energy. But there is a problem. When 3 number phosphate is attached at glycerate, this is not a high energy bond. This is not a very high energy bond. If I break this bond here, then I won't be able to get enough energy to make ATP, right? So what we have to do is that first we have to make this bond high energy and then we will break this last high energy bond. So our next 2 steps, that is number 8th and number 9th step, is like a preparation to make high energy bond. So let's see the detail. 3-phosphoglycerate gets converted to 2-phosphoglycerate. So phosphate is getting transferred from number 3 position to number 2 position. And the name of enzyme is mutase. So what is the EC number of mutase? Transfer of phosphate is occurring from position number 3 to 2, but mutase is not EC number 2 transferase. No, it is not transferase, because in this step molecular formula is not changed. One isomer is getting converted to another isomer, transfer is occurring, but within the molecule, intramolecular transfer is occurring, and whenever in cells intramolecular transfer is occurring, be it methyl group, sulfate group, phosphate group, the enzyme used is always mutase, and mutase belongs to isomerase category, that is EC number 5, not EC number 2. Be careful with this. Now next step, 2-phosphoglycerate gets converted to phosphoenol pyruvate. Enol. Focus here. En means double bond, ol means OH. So this kind of bond with double bond and OH is made here. So you can just take an idea that this is a high energy bond. And to make this enol high energy bond, which enzyme is used? Enolase, right? So to make enol bond, it is enolase enzyme, very easy, right? Now, phosphoenol pyruvate we have obtained. That is, we have our last phosphate attached to pyruvate with high energy enol bond. We can break this enol bond and we can get pyruvate. On the other hand, phosphate is taken by ADP, gets converted to ATP. I have written 2 here because we started with 2 molecules. Whenever ATP formed at such a step known as substrate level phosphorylation step, the enzyme must be a kinase, and here it is pyruvate kinase. Here it is pyruvate kinase. Now pyruvate is the end product of aerobic glycolysis, but lactate is formed in case of anaerobic glycolysis, and here the enzyme is lactate dehydrogenase, short form LDH I have written, lactate dehydrogenase. Can you tell me where is pyruvate dehydrogenase? Pyruvate dehydrogenase is in link reaction, right? But this lactate dehydrogenase is in anaerobic glycolysis. So we are done with the pathway and all the 10 to 11 enzymes we are done. Now I'm going to write the EC numbers of all the enzymes. See, hexokinase EC number is 2 because it is a kinase. Isomerase EC number is 5. Then PFK1, it is a kinase, so EC number 2. Aldolase A, it is breaking the bond without using water. That's why EC number is 4, right? 4 is lyase. Bond is made or broken. Water and ATP not used. So aldolase A or aldolase B both are in EC number 4 category. Then 5th number enzyme is isomerase. So EC number is also 5. Then 6th number enzyme is dehydrogenase. Dehydrogenase means EC number 1. Then PG kinase, so 2. Mutase, 5 I told you. Enolase is EC number 4. This I will explain later in enzymes topic. Then pyruvate kinase, because it is a kinase, so EC number 2. And the last one, lactate dehydrogenase of anaerobic glycolysis, EC number is 1 here, right? Now there are 3 irreversible steps in glycolysis. These are, you can see here, broad black arrows I have made. These broad black irreversible arrows I have made. So these three enzymes, these three steps are irreversible or regulatory steps of glycolysis, that is hexokinase, PFK1 and pyruvate kinase. So you can note here that all these three are kinases, right? Hexokinase, pyruvate kinase, phosphofructokinase. So these three regulatory steps are kinases. Now there are total 4 kinases in glycolysis. One extra is PG kinase. So can I say that all kinases of glycolysis except phosphoglycerate kinase are irreversible or regulatory steps of glycolysis? And now let's see the substrate level phosphorylation enzymes of glycolysis. That is this and this, that is PG kinase and pyruvate kinase. They are substrate level steps of glycolysis. Now you can see pyruvate kinase enzyme is coming in the list of irreversible regulatory enzymes also, and pyruvate kinase is also substrate level phosphorylation enzyme. This enzyme is common in both the list. I told you something special about pyruvate kinase enzyme. You have done that. Pyruvate kinase enzyme requires potassium more than magnesium. Most of the kinases they use magnesium, but pyruvate kinase requires potassium more than magnesium. Now one more point about pyruvate kinase is that this enzyme deficiency is the second most common human enzyme deficiency. There is one more enzyme which is the first most common human enzyme deficiency, that is G6PD enzyme, which is enzyme of HMP pathway. Pyruvate kinase enzyme is of EMP pathway or glycolysis. G6PD is enzyme of HMP pathway, and this is the first most common human enzyme deficiency. Now both these patients, that is pyruvate kinase deficient and G6PD deficient patients, they both are very common. See, first most common and second most common enzyme deficiency. They are common and they have one common clinical feature, that they both present with hemolysis. So how will you distinguish between these two patients? There's a simple thing, Heinz bodies. You know, Heinz bodies are present in G6PD deficiency. So if hemolysis and Heinz bodies are present, then it is G6PD deficient patient. If hemolysis is present but absent Heinz bodies, then most probably it is pyruvate kinase deficiency.